Hello, everyone. In our today's lecture of medical microbiology, we are going to learn about the pathogenic bacterial species in which our today's opportunistic pathogen is Acinetobacter. Now, this Acinetobacter, the name Acinetobacter came from the Latin word for motionless. As Acinetobacter are non-motile, they are not able to move from one place to another due to absence of cilia or flagella. They are motionless. So, these organisms were known as Acinetobacter, and there are near about 32 species of Acinetobacter, and in which Acinetobacter baumannii and Acinetobacter lophi have the greatest clinical importance, as they are opportunistic pathogen and have the ability to cause the infection. So in the clinical field they have the great interest of study. The introduction to Acinetobacter. Now, most species are more not significant source of infection. Now, generally, all the species included in Acinetobacter does not cause the infection, but few species like Acinetobacter baumannii are opportunistic pathogen and have generally found in the hospitals. There is high risk to the people who have suppressed immunity or the less immunity, have the high risk of contamination or the cause of infection by the Acinetobacter baumannii. Now we will see the few morphological characteristics of Acinetobacter. Now, Acinetobacters are gram-negative bacteria possessing cocobacilli shape, and they are strictly aerobic in nature, and they are non-motile organisms. As we have seen in the first slide, they are motionless bacteria which does not possesses cilia or flagella, so they are non-motile. If we study the biochemical nature of Acinetobacter, they are catalase positive and oxidase negative. So next one is the epidemiology of Acinetobacter. They are generally found in the environmental reservoirs, means they are generally ubiquitous in nature. They are found in soil, fresh water, vegetables, animals, even in the body lice or fleas and ticks also the Acinetobacters are found. And in hospital environment, generally in the environmental surfaces, and the patients coming in the contacts with the various equipments used in the hospitals like ventilators, dialysis machines, air ventilation system, water sources, etc., are highly contaminated by the Acinetobacter. Even if the person comes in the contact with the surface, like touching the surface with the hands or the contaminated suction equipments, there also the Acinetobacters can be present. They are also present in the respiratory tract, urinary tract, gastrointestinal tracts, and wounds of the different patients. These bacteria may be present. Now, what are the requirements, or what type of conditions are required for the growth of Acinetobacter? Now, these organisms are strictly aerobic, so proper oxygenated conditions are required for their growth. They generally grow at the temperature 44 degrees Celsius, and the media required for the growth of Acinetobacter is differential media is McConkey agar. On the McConkey agar also they can grow, and the selective media used for the growth of these organisms is chrome agar or leads Acinetobacter agar. Now here are the few pictures in which you can see the growth of Acinetobacter on the various medium, which is your Meconic agar, leads Acinetobacter agar and the chrome agar. Next point is biochemical profile of Acinetobacter. As we have seen earlier also, both Acinetobacter baumannii and Acinetobacter lawfii are catalase positive and oxidase negative. If we see the fermentation properties, Acinetobacter baumannii ferments different types of sugars like glucose, xylose and lactose, but Acinetobacter lawfii cannot carry out the fermentation process. How the presence of Acinetobacter can be detected? Now the presence of Acinetobacter in various infection or on the surfaces can be rapidly detected by using various various kits like RAP-ID1 panel and API 20E strips. This can be used to check the presence of Acinetobacter at the specific levels. In diagram you can see those panels which are used for the estimation or the presence of Acinetobacter. The molecular detection of these organisms can be detected by PCR technique or polymerase chain reaction, where the specific primers like Rec-A is used to detect the Rec gene in Acinetobacter baumannii, and by using these specific primers in PCR technique, easily the presence of different species of Acinetobacter can be detected. Now the pathogenesis or the ability of Acinetobacter to cause different diseases are, now they are generally opportunistic pathogens, and they survive under the dry condition, as the Acinetobacter requires high temperature for their growth, that is 44 degrees Celsius of temperature, and dry conditions are required or necessary for the growth of Acinetobacter by coming in contact with the patient or the surface infected or contaminated with the Acinetobacter. They can cause the infection easily. Now what are the virulence factors or the how they can cause the infection to the any particular person? After the entry of Acinetobacter, a polysaccharide capsule is formed which prevents the activation of complement factors. If the complement activation does not take place, the different cells or the immune response cells does not get activated, and the phagocytosis process is avoided. As the phagocytosis process is avoided and complement system activation does not take place, these Acinetobacter or opportunistic pathogen can easily survive inside the human body and cause the infection. In this, the fimbriae helps in the adhesion or attachment of Acinetobacter to the human cells, and pila helps in the colonization on the surface of different cells and form a biofilm of Acinetobacter and cause the infection. Now how this Acinetobacter or the infection caused by this organism can be transmitted from one person to another person or from the patient to the healthy person. Now, it can simply be spread from person to person by coming in contact or exposure to the surface which is contaminated with this organism. Simply, if you comes in contact with the patient suffering with infection caused by Acinetobacter, or if you touch or you come in contact with the surface which is contaminated with this organism, you can get infected with the infection. Now, the antibiotic resistance of Acinetobacter. Now, generally, these Acinetobacters are multi-drug resistant organisms. These organisms have the ability to accumulate the multiple antibiotics, and resistant genes to these antibiotics are present inside the Acinetobacter, which leads in the development of multiple drug resistance of these strains. Means if they are unaffected or resistance to various different types of antibiotics, so they can easily survive for the larger time inside the cell. Now, how does multiple drug resistance is developed, or by which mechanism does the Acinetobacter develops the antibiotic resistance? These Acinetobacter have the antibiotic altering enzymes. They alters the structure of the antibiotics, or they can even cause the mutation in themselves, which leads to produce the resistance against those antibiotics. Now, which type of treatment can be supplied to cure the infection caused by Acinetobacter? Now, as we have discussed earlier, that they are multiple drug resistance, and this is the big problem for the treatment of the disease caused by the Acinetobacter. Now, generally, Acinetobacter baumannii is a big problem in many hospitals. In different types of hospitals, the infection caused by Acinetobacter is a major problem. Generally, if the infection is caused by this species, the first line of treatment is done with the help of carbapenem antibiotic. Now, carbapenem antibiotics such as imipenem is generally used for treating the infection caused by Acinetobacter. But nowadays, there is increase in the resistance of the Acinetobacter towards the carbapenem. So, other treatments or other antibiotics are also used as a treatment for the infection caused by Acinetobacter are polymyxin, then tigecycline, or aminoglycosides are the different types of antibiotics which are used for the treatment of the infection caused by Acinetobacter baumannii. Now, how the resistance or how the treatment to the resistant infection can be done. Now, colistin and the polymyxin B antibiotic have been used to treat highly resistant Acinetobacter infection. If at the first line, if imipenem does not helps to cure the infection, then the colistin or polymyxin B antibiotic is used for the treatment of this infection. But the choice of appropriate therapy is also a complication, because uh, cholesterol is slightly toxic to the renal system of the patient. If the cholesterol is given to the any particular human being or the patient, it can cause the toxic effect to the patient. So generally cholesterol is not used at larger scale. And nowadays, so few species of Acinetobacter are also isolated, which have even developed the resistance to these antibiotics of colistin and polymyxin B. So this was all about the morphology, pathogenesis, epidemiology, and treatment, and the biochemical nature, etc., about the Acinetobacter. So in short, Acinetobacter is the opportunistic pathogen which can cause the infection. Their nosocomial infections are caused by Acinetobacter. They grow best at the aerobic condition, can be transmitted by the contacts. They possess antibiotic resistance, so the treatment is slightly difficult. So, how we can get rid of these Acinetobacter? Just by washing your hands and shut off them. Prepare, maintain proper hygienic conditions, and in now the pandemic situation also, prepare maintaining proper social distancing and a proper hygienic condition, continuously washing hands can keep you safe from the different types of germs and as well as the infection caused by Acinetobacter. So this was all about the Acinetobacter species. Thank you.